so hopefully everybody is looking at my screen. Hopefully everybody can see the notes. This is the last part of the notes from 14.2. We are going to be talking today about the transcription. Now, we've been talking about transcription and translation and protein synthesis for a minute now, but this is going to formally cover that topic. So if you are looking at your note catcher, this should be the very top of page 3. So take a minute, write this down, and then we'll talk about it, okay? 1 minute, write it down, and then we can talk. All right, so again, we've been talking about protein synthesis. Protein synthesis is the process by which our DNA gives instructions for proteins to be made. Again, why do we care? Because proteins are what makes life possible. They are the embodiment of your traits. If you have brown eyes or blue eyes or hazel eyes, if you are or are not lactose intolerant, if you have, if when you eat cilantro it tastes like dirty soap, or if it tastes delicious to you, these are all traits, and they are all controlled by proteins. Protein synthesis takes place in 2 steps. We talked about transcription, which is the process of actually building the mRNA from the DNA in the nucleus. And then we said the mRNA goes out to the ribosome to actually, build the protein. That is the process of transcription, which we are going to be talking about today. So, genetic coding takes place in many, many steps. Step 1, we need to understand that our amino acids, remember, which are the building blocks of proteins, when we first string them together, they are in very, very long chains called polypeptides. They are called this because the bond between the amino acids are called peptide bonds. So polypeptide literally means many of these bonds. Specific amino acids in a polypeptide and a specific order and a specific folding make a specific protein. That creates the unique properties of proteins, which then can create your unique traits, or your cat's unique traits, or your dog's unique traits, or a tree's unique traits. So it is the sequence, what order those amino acids show up in, which again are going to influence the shape of the protein, which in turn determines the function, what the protein actually does. Remember, there are 20 different amino acids, but how we put those amino acids together is what determines the protein. Yeah, it doesn't sound like that many, but from those 20 amino acids, we can literally build millions of proteins per individual organism on this planet. Issues or questions? Hearing none. All right, quick review. Going back to RNA base pairing. RNA has 4 different bases. And again, this is based off of Chargaff's rule. Adenine pairs with uracil, cytosine pairs with guanine. We know this. We've done this already. These base pairs are called the genetic code. The code reads in 3s. We know this also. These 3-letter word combinations, or 3-letter combinations, are codons, and each individual codon, like we did this morning in our warm-up, corresponds to a specific amino acid. So this morning we did UUG, and we found that UUG led to the amino acid leucine. Okay, so UUG calls up leucine. There are several combinations that will lead to the same amino acid, just so that you know. Kind of like in math, like you can say like 3 plus 4, or I'm sorry, like 3 plus 1 is 4, but 2 plus 2 is 4, but 5 minus 1 is 4. 2 times 2 is 4. So you have several different ways of getting to the same place. The same thing is true with amino acids. There are a couple of different codons that may lead to this to the same amino acid, but the important thing is that you know how to actually read that chart that we took a look at this morning. Again, because these codons are going to call up these specific amino acids, and the order of those specific amino acids is going to determine the protein, and the protein is going to determine the trait. Questions or issues? All right, hearing none. All right, so there are 4 different possible bases, and there are 3 in a codon. So 4 times 4 times 4 is 64. What this means is there are 64 possible codons mathematically. Like that's just math. But again, most amino acids are specified by more than one codon. We know this. We just talked about this. And again, like I said, this is the same thing like in math, how like 2 plus 2 is 4, but 3 plus 1 is 4. 5 minus 1 is 4. You can get to the same place with different combinations, and that's okay. Because again, there's 64 possible combinations, but there's only 20 codons. So there has to be some overlap. Like here it says, for example, there are 6 different codon combinations for leucine, but there's only one combination for tryptophan. So there you go. Like again, sometimes you have multiples, sometimes there's only one way to get there. Now, remember, when we talked about codons originally, we said that there were these things called start codons and stop codons, and that they were a specific chemical signal that would tell the rRNA, hey, like start coding here. This is the beginning of my protein. And then the stop codon is the end of the protein. The most common start codon is AUG, and AUG codes for methionine. Okay, so AUG or methionine is our start codon. And again, we hopefully know how to do our decoding based on the genetic code chart. And again, there is the square one, there is a circle one. My personal preference is the circle one, but if you like the square one, I'm not going to stop you from using it. Okay, so codon charts are used to code for amino acids. We already know how to do this, but again, quick review. You start with the first letter of the codon in the middle circle. Then you move out to the second ring of the second letter, third ring, final letter, and then read the codon. So remember that methionine is A, start in the middle, A, go to the next ring, U, go to last ring, G, methionine, AUG is methionine. And again, methionine is our start codon. Now, this is very similar to the video that we saw, I want to say on Friday, maybe Thursday. Shouldn't take too long. And we're not going to watch the whole thing. I just want to go over the part where they're going over the codon chart.
Check out this verbo.
Oh man.
Oh,
They're your cousins.
Look at me. Come on, here.
I want to show you how to use this new type of code.
give me 1 second. Let me go to full screen. It won't let me go. There it goes. It's just really laggy today. There we go. All right.
Codon chart that you haven't seen before. I'm just going to teach you how to do the first one, and then you can do the rest on your own. So basically all you have to do is you look at the first letter of the codon. In this case, it's A. So you start in the middle. You find the A. It's right here, right? The next letter is U. So from here, you need to find the U that's attached in the A quadrant, right? So it's everything that's here, everything that's touching this A, right? So the second one is U right here. The last one is G. So then you've got to find G. CUG is right here, and it codes for MUT, which, guess what? That is our start codon. Makes sense, right? So, first thing you do, go to the middle, find your first letter, then go to the next line and find your second letter, depending upon where your first letter is, and then go out and find your last letter, and then you'll find your amino acid out on the end.
All right, so let's look at this last one, which I imagine is coding for a stop codon, but it might not be. So if we take a look at UAG, start up here, U, first letter, second circle, A, third letter, third circle, G. AUG is a stop codon. Again, no big surprise there. You start with a start codon, you end with a stop codon. Now, methionine, which is our start codon, there's only one, but I do want to point out UAA is a stop codon, UAG is a stop codon, and UGA is a stop codon. So we do have these three stop codons over here in the U quadrant. All right, any issues or questions on how to use a codon wheel? All right, fantastic. All right, now sometimes we have abbreviations, sometimes we have the full names. The ones that I like to use have them both. Again, if you choose to use the abbreviation, use the abbreviation given in the chart. Do not make up your own, okay? But the chart that I will be providing you will have the standard abbreviations, if you will. All right, so this is on your paper. So let's take a quick look. And I am going, let me bring up the codon wheel from earlier and let's see if somebody can help me out. If I have GAC, and like I said, I'm going to open the codon wheel from earlier. That was a little easier to look at. All right, so GAC. Who can tell me what GAC is going to code for? GAC. Who can tell me what GAC is going to code for?
I did not know I said that, but ASP. Yeah, I
G.
know it started with an ASP though.
Aspartic acid.
Yeah.
There it is. Okay, GAC, aspartic acid. Or if you prefer, just ASP. Okay? Again, you can use abbreviations if you want. All right, second one in your notes. Who can tell me what AAG codes for? AAG. Thank you. Somebody's in the chat. Lysine. Excellent. A, start in the center, go out to the next one. A, G, lysine. Perfect. What about, okay, who can tell me what's wrong with what I did in the next one? Because that is just messed up. What is wrong with the next one? Why does TCC wrong?
Because ain't no T.
Yeah, there's no T in RNA. I should have written UCC. So let's see if we can figure that one out. It should be UCC. That is a typo on my end. Who can tell me what UCC is? All right, it's in the chat. Oh, 3 people in the chat. Good job. Serine, perfect. You here in the center. Serine, or SER. All right, who can tell me what ACA is? ACA.
THR.
THR, or threonine. And I
Yep.
had 2 people in the chat say that too, so yay you. ACA. Threonine or THR, whichever you prefer. And then, of course, that last one, we were going to have to substitute in the U again. Obviously, I was thinking DNA, not RNA, so shame on me. What is AUC? Get a code for AUC. Okay, yep, there it is. I got a couple people in the chat. AUC is isoleucine, or ILE. AUC, isoleucine, or ILE. Awesome. So thank you to everybody who participated doing that. I appreciate you. All right. So we already kind of did that, so we can just go ahead and move on. We already did this part. Start and stop codons. Here we go. All right, so your start and stop codons, they're pretty much like punctuations in a sentence. So, much like a period ends a sentence and a capital letter starts a sentence, that's kind of like what these start and stop codons do. They start coding, they can pause coding, kind of like a comma, they can stop coding. As we already said, there are 3 different stop codons that end translation. Remember, translation is the part of protein synthesis where we're actually going to be building the protein. So UGA, UAA, and UAG are all stop codons. When you have a UGA, a UAA, or a UAG, when you have a stop codon, then that's when translation stops. In other words, the polypeptide chain is complete, and it is now time to fold that polypeptide chain in a very specific way to make that very specific protein for your very specific trait, whether that's your eye color or your ability to, digest milk or whatever it is. All right. Issues or questions? All right, so we are now, actually, we already had started the 5th page, but that's our second. I don't even know. I know your guys' handout's a little bit different than mine. All right, so sequences and nucleotide bases are the instructions that determine what the amino acids should be and what order they should be when they produce a polypeptide. But again, for that polypeptide to actually become a protein, we have to shape it. We have to put it into a final fold. We have to put it into a final shape in order for the protein to be assembled correctly. It's kind of like buying furniture at IKEA, some assembly required. So once a completed polypeptide is folded into its final shape, or maybe it joins up with another one and then they fold up together, then and only then does it become a fully functioning protein. Then and only then can that protein then go out and do its job in creating that trait for you or the offspring. So again, this is no different than if you've ever had to put together Legos or any other kind of like put together like model or anything like that. Furniture, if you've ever gotten some furniture from IKEA and had to help your folks put it together. Okay, so that is what this is very similar to. You have a bunch of pieces, but you gotta put it together. And you have to put it together properly if you want the protein to function properly. All right, so who is going to do all this assembly for us? The assembly is done in the ribosomes. Again, we know this, we've talked about it, but now we're going to talk about it in a little bit more detail. Ooh, computer's acting funny. All right, so again, if you've ever had to assemble a toy, if you've ever had played with Legos, if you've ever had to put together furniture from IKEA, sometimes maybe, hopefully, sometimes you've always read the instructions. Sometimes the instructions aren't awesome. In which case, you have to kind of go in and use some of your prior knowledge. So what happens once we have the instructions? Who is going to put this together properly in the cell for this protein? Ribosomes. Remember, ribosomes is made up of rRNA. Remember, the RNA is our doers. So the mRNA brings the instructions, but the rRNA is actually going to do the work of actually assembling the actual protein itself. So the ribosomes use the mRNA instructions to assemble the amino acids into the polypeptide chain. They decode the mRNA message, and the rRNA, the ribosomes are actually the most important piece in translation. They translate the message to create the protein. Issues or questions? All right. Transcription, translation, where do they happen? Remember, protein synthesis is 2 steps. Transcription happens first. It's not a part of translation, but we need it first. Then after transcription, then we have translation. Remember, transcription happens in the nucleus because transcription is the process of making mRNA, which is pretty much just like a copy of your DNA. So we're copying the instructions. Translation is the actual building of the proteins. Translation happens in the ribosomes, which means it's happening in the cytoplasm. So transcription happens in the nucleus, translation happens in the cytoplasm. All right, we've talked about this before, ladies and gentlemen. All 3 of our RNA molecules are going to work together in order to translate genes, okay? So your mRNA brings in the message. The tRNA goes and fetches the amino acid in the cytoplasm, and the rRNA uses the message from mRNA and the amino acids that tRNA brings over, and it then will use both of them to assemble the final message, the final protein, the final polypeptide chain. Okay? So mRNA is the message, tRNA delivers the amino acids, and again, it's going to have to read the codons to do that, and rRNA helps hold together our proteins to help bind and hold together our proteins. Again, none of this should be brand new to you. We've talked about this for the last almost 3 weeks at this point. Okay, so the translation process happens in 5 steps, okay? Step 1, mRNA is transcribed in the nucleus and enters the cytoplasm. That's transcription. That's not even really part of translation, but whatever. Step 2, the mRNA leaves the nucleus and then goes out into the cytoplasm and attaches to the rRNA, attaches to the ribosome. Step 3, the tRNA starts bringing in, transferring in the correct amino acids into our rRNA, our ribosome. Okay. Step 4, our polypeptide chains begin to grow, they begin to form until we reach that stop codon that we had just talked about. Finally, when we get to that stop codon, we can release the chain, and then that chain gets folded, maybe hooks up with another polypeptide chain, they fold together, and then we release it into the cytoplasm, and now we have a protein. So again, protein synthesis is a 5-step process. Step 1, that's transcription. Step 2, 3, 4, 5, that is translation. So translation is a little more intense. All right, let's take a quick look at our overview of protein synthesis.
So I've got my essay written, and I've been working on it for about a week. So now I'm going to show you how I use Grammarly to edit. Okay, so here's my essay, and I'm going to click through Grammarly's suggestions in the right panel.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So the small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell, wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then, the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. and that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first three letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column, and the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and follow me on Twitter at SciencePet.
All right.
Yeah, I'm gonna call it right now. I think I found the single biggest productivity hack of 2021. It's this Google Chrome extension called Speechify, and it literally does my reading for me.
You will not be allowed to take a class, and you will be swept down to the media center, where you will be giving a warning if it is your first time late to class. You will remain there. You're not going to get a hall pass.
All right, ladies and gentlemen, we are almost to the end of our notes. We probably won't get till Central Dogma until tomorrow, but that's okay. That should only take us a few minutes. So, again, let's take a quick look at the beginning, middle, and end of protein synthesis. 1 second.
You're not just going to, again, get warning after warning. Please go to class. You will be sent home, and you must have a parent return with you to readmit you to Oak Park High School. Again, this is a consequence of your actions of not getting to class on time and looking for different ways to avoid that. So again, ladies and gentlemen, please make sure that you are moving when the bell rings to your second hour. Again, do not come down to the Welcome Center. Do not come down to the main office. There are no passive security, and other staff members will escort you to the media center, where you will remain, cell phone taken or given to you, so that, again, you can work on your skills. If you don't want to go to your classroom to work on your skills, you can work on your skills in another area. Again, after 2 times of that happening, again, there's no more warnings, there's no more talking, there's no more excuses. You will be sent home, and again, you must have a parent come back with you to get back into Oak Park High School. Get to your classes on time. Thank you.
I apologize. They were doing an announcement here, and I had to listen to it. Oh, my goodness. And it's just about that time anyways. So we're going to actually pause here. Let me turn off my video, and we will continue this tomorrow.